Hello everyone. Now we reached the chapter 8. The topic of this chapter is monoprotic acid-base equilibria, where the acid-base equilibria or chemistry is the core of analytical chemistry. The author of this book, Daniel Harris, recommends that if you don't have enough time to teach all the content in the book, you choose the acid-base chemistry. Okay, acid-base chemistry is really important in not only in chemistry but also in other subjects. Okay, but I found something rather strange, which is that many students think they have enough knowledge of acid-base chemistry, but actually, um, as you know, there is a very deep things about acid-base equilibria. This is not simple matter, okay? Especially when you deal with a weak acid and base equilibria, and also there are some polyprotic acid and bases, okay? So this chapter is very important. So after you learn this chapter and the following few chapters, you will have deep knowledge of acid-base chemistry. Okay, let's start. The front page of this chapter shows the measuring pH inside cellular compartment. Okay, since the pH is defined by the negative logarithm of proton concentration, and pH plays a very important role in chemical reactions in aqueous solution. Okay, the pH measurement is also very important. Usually, pH measurement is done by pH meter. Okay, um, the pH meter uses usually glass electrode as a pH sensor. Of course, there are different types of pH sensors, but they are all big, much bigger than the size of a cell. Okay, but question is, how do you measure the pH inside these cellular compartments? Okay, and sometimes we need to know the pH inside the cell. Okay, this is not very simple matter, right? Because you cannot insert your pH sensor into the cell because there is such a big difference in sizes. So you need to have another other technique to measure the pH inside the cell. Okay, this example is about that. This first picture is the picture of this one, mouse macrophagy. Okay, eating these two foreign red blood cells. Okay, this process is called phagocytosis. The phago means eating. Okay, it has a Greek origin. Phagein means to eat. Okay, cytosis means cell. Okay, this is a cell eating. As you know, um, when a foreign species enters into our body, and then our immune system works. So this macrophage is a white blood cell, and it fight infection by ingesting and dissolving foreign cells. Okay, the process is called phagocytosis. Okay, in this case, see, uh, you may want to know the monitor the pH change inside the cell. Okay, how can you do that? Okay, one way is to use um, the fluorescence measurement. Okay, here this second picture shows that two macrophages. Okay, and it has small bead inside. That bead is 1.6 nanometer, 1.6 micrometer diameter fluorescent beads. Okay, now it uh, ingested this fluorescent bead. Okay, so this one, the panel C shows the fluorescence image of this panel. Okay, yeah. Okay, let's consider this macrophagy. Okay, macrophagy, a macrophage, I'm sorry, modules with lysosome containing digestive enzymes that are most active in acid. Okay, actually, uh, this lysozyme, okay, is merged with this macrophage. This lysozyme, um, in Korean, it's a sopoche. It contains the enzymes that digest, okay, foreign species. Okay, the lysozyme is an organelle in the cytoplasm of eukaryotic cells containing digestive enzymes enclosed in a membrane. Okay, the important is that this enzyme activity is most active in acid, not in neutral or in basic conditions. Okay, so it is important to measure the pH inside the cell. Okay, one way is to use fluorescence. Okay, one is to measure pH in lysozyme to present macrophage with polystyrene bead with fluorescent dyes. Okay, these small, the spheres are made of polystyrene bead with fluorescent dyes. Okay, what kind of fluorescent dyes can be used? Okay, there are two types of fluorescent dyes. One is this one, fluorescein. Okay, this a rather big molecule. It has a property of emitting fluorescence. Okay, and the other one is tetramethylrhodamine. This one also fluorescence. Okay, uh, emits fluorescence. What difference is that this fluorescein molecule, the fluorescence intensity quite depends on pH. Okay, but this one, this species fluorescence intensity, does not change with pH. Okay, this one. Look at this figure, and this is the fluorescence uh, spectra depending on different pHs. Okay, and it has two peaks, one at this wavelength, okay, around 530, okay, or 2 or something nanometers, and the other one is 5 maybe 75 nanometer. Okay, this peak belongs to this fluorescein molecule, and this peak depends on tetramethylrhodamine. Okay, at higher pH, okay, this fluorescence intensity of fluorescein uh, is highest. Okay, this is fluorescein emission. This depends on pH, and as pH increases, as acidity of the solution becomes more and more acidic, and this fluorescent intensity gets smaller and smaller. But this peak doesn't change much. Okay, so by taking the ratio of these two peaks, we can estimate the pH inside the cell. All right, that's the principle. Here, look at that. This figure shows the changes in fluorescence intensity ratio. Okay, which means the fluorescein, and rhodamine, the ratio of this peak and this peak. Okay, as a function of time. Okay, when this macrophage, okay, eats the foreign red blood cell, and then the the enzymes, okay, digestive enzymes in lysozyme works. Okay, and it works at the lower pH. Okay, so the inside the pH changes around the pH 7.3, okay, around here, and then as time goes by, the pH suddenly drops. Okay, so it remains at pH 5.7. Okay, which means that um, now the system is working fine. Okay, and system, the internal pH becomes more acidic, so that the digestive enzymes are working active. Okay, yeah, this small illustration, it shows the importance of measuring pH, in this case inside the cell. Okay, so now we study one by one. Okay.